Welcome to SS2 Biology, Term 3, Week 4. Biotechnology uses living organisms or biological systems to develop products and processes that benefit humanity. In the 21st century, genetic engineering, the direct manipulation of DNA, has become one of the most powerful and controversial tools in science. By the end of this lesson, you will define biotechnology and genetic engineering, describe key techniques including recombinant DNA and CRISPR, explain applications in medicine and agriculture, discuss ethical issues, and apply this knowledge to WAEC examination questions. Let us begin. Biotechnology is defined as the application of biological systems and living organisms to develop or make products useful to humans. Traditional biotechnology has been practiced for thousands of years: fermentation, bread, beer, wine using yeast Saccharomyces cerevisiae, oji, pito, burukutu in Nigeria using wild yeast and bacteria, cheese and yogurt production using lactic acid bacteria, antibiotic production, penicillin from Penicillium mold discovered by Alexander Fleming in 1928. Modern biotechnology began in 1973 when Herbert Boyer and Stanley Cohen created the first recombinant DNA molecule. Key techniques: recombinant DNA technology. A desired gene is cut from a donor organism using restriction enzymes, molecular scissors that cut DNA at specific sequences. The gene is inserted into a vector, usually a plasmid, a small circular DNA molecule from bacteria. The recombinant plasmid is introduced into a host cell, bacterium, yeast, or mammalian cell. The host cell expresses the gene and produces the desired protein. Polymerase chain reaction. PCR amplifies a specific DNA sequence millions of times in a few hours using cycles of heating and cooling, invented by Kary Mullis 1983, used in COVID-19 testing, forensics, paternity testing, diagnosing HIV and malaria in Nigeria. Biotechnology has transformed medicine. Recombinant insulin. Before 1982, diabetics used insulin extracted from pig and cow pancreases, expensive, allergenic, and limited in supply. In 1982, Genentech and Eli Lilly produced human insulin using recombinant E. coli bacteria containing the human insulin gene. Today, all insulin used worldwide is recombinant human insulin, cheaper, safer, and infinitely scalable. Nigeria has over 4 million people living with diabetes. Insulin access remains a challenge. Hepatitis B vaccine. The HBsAg, hepatitis B surface antigen, gene is inserted into yeast. Yeast produces the antigen protein. This protein is used as the vaccine. This was the first recombinant vaccine, 1986. Nigeria has among the highest HPV infection rates in the world. Approximately 15% of the population is HPV positive. Vaccination at birth is now standard under the National Program on Immunization, NPI. Erythropoietin, EPO. Recombinant EPO stimulates red blood cell production, used to treat anemia in kidney patients and cancer chemotherapy patients, also abused in endurance sports as a doping agent. Monoclonal antibodies, identical antibodies produced from a single B-cell clone, used in cancer treatment, trastuzumab for HER2+ breast cancer, autoimmune disease, infliximab for rheumatoid arthritis, and diagnostics. Pregnancy test strips use monoclonal antibodies against HCG. CRISPR-Cas9, clustered regularly interspaced short palindromic repeats. A bacterial immune system adapted as a gene editing tool. Doudna and Charpentier, Nobel Prize 2020, more precise, cheaper, and easier than earlier gene editing tools, can correct genetic diseases by editing DNA in living cells, used in sickle cell disease trials. In 2023, the FDA approved the first CRISPR therapy, Casgevy, for sickle cell disease and beta thalassemia. This is profoundly significant for Nigeria, which has the world's highest burden of sickle cell disease. Genetically modified organisms, GMOs, are organisms whose genome has been altered using genetic engineering. Bt cotton and maize, the gene for Bacillus thuringiensis, Bt toxin, a naturally occurring insecticide, is inserted into crop plants. Bt plants produce their own insecticide, killing specific pest insects while remaining safe for humans and other animals, reduces pesticide use. Nigeria approved Bt cowpea in 2019, the first GM crop approved in Nigeria. Cowpea, black-eyed peas, is a critical protein source for Nigerians. The pod borer, Maruca vitrota, destroys up to 80% of unprotected crops. Bt cowpea carries a resistance gene, dramatically increasing yields. Golden rice engineered to produce beta-carotene, provitamin A, in the grain, designed to combat vitamin A deficiency, which causes blindness and death in approximately 250 million children globally. Vitamin A deficiency is common in northern Nigeria. Herbicide-tolerant crops. Genes from herbicide-tolerant bacteria introduced into crop plants, e.g. Roundup Ready soybeans tolerating glyphosate herbicide, allows herbicide application that kills weeds without harming the crop, increases yield but raises concerns about herbicide-resistant superweeds. Disease-resistant bananas. Panama disease, Fusarium wilt, threatens banana production worldwide. GM bananas with resistance genes are in development. Benefits of GMOs: increased crop yields, reduced pesticide use, drought and flood tolerance, enhanced nutritional value. Controversies: ecological risks, gene flow to wild relatives, effects on non-target insects, socioeconomic concerns, corporate control of seed supply, farmers cannot save GM seeds, regulatory gaps in Nigeria. Biotechnology raises profound ethical, legal, and social questions. Genetic privacy. Genomic testing reveals information not only about the individual but about biological relatives. Who has the right to this information? Insurance and employment discrimination based on genetic predispositions is banned in many countries. Gene therapy versus genetic enhancement. Gene therapy corrects a disease-causing mutation. Broadly accepted, genetic enhancement modifies traits in healthy individuals. Enhancement of height, intelligence, athletic ability, widely considered ethically unacceptable. Human germline editing. Editing the DNA of embryos, eggs, or sperm creates heritable changes affecting all future generations. In 2018, Chinese scientist He Jiankui shocked the world by editing embryos to be HIV-resistant, creating the first gene-edited babies. He was widely condemned, sentenced to 3 years imprisonment in China, and his experiment was considered premature, risky, and unethical. GMO concerns. Many African countries including Kenya initially banned GM crops pending safety assessment. Nigeria's National Biosafety Management Agency NBMA, established by the Biosafety Act 2015, regulates genetically modified organisms. The Cartagena Protocol on Biosafety 2000, ratified by Nigeria, governs transboundary movement of GMOs. Religious and cultural concerns about playing God or interfering with nature are important to respect in diverse societies like Nigeria. Key points: biotechnology is the use of living organisms for human benefit, traditional, fermentation, antibiotics, and modern, recombinant DNA, CRISPR. Key techniques: recombinant DNA using restriction enzymes and plasmid vectors, PCR for DNA amplification, CRISPR, Cas9 for gene editing. Medical applications: recombinant insulin, HBV vaccine, EPO, monoclonal antibodies, CRISPR sickle cell therapy. Agricultural applications: Bt cowpea, Nigeria 2019, golden rice, herbicide-tolerant crops. Ethical issues: genetic privacy, germline editing, GMO regulation, Nigeria, NBMA, Biosafety Act 2015, Cartagena Protocol. WAEC biology questions on biotechnology require knowledge of specific techniques, named products, and their benefits and risks.